What are communicating junctions and how do they work? Another name for communicating junctions is gap junctions, and gap junctions are formed by a complex of connexons proteins that are going to be embedded within the membrane. They're going to be an example of a transmembrane protein. And basically, we've got a series of these individual proteins coming together to form this gap junction complex. It's going to have a closed configuration and then a shape change, which is going to result in an open configuration. And with that open configuration, basically what we're going to end up with is going to be a hydrophilic pore that is going to extend from the cytoplasm of one cell, kind of represented on the top portion of the slide here, pass through both of the cell membranes, so pass through the hydrophobic barrier. But then we've got this, in essence, tunnel, this hydrophilic tunnel for materials from the cytoplasm of one cell to flow through this gap junction and into the cytoplasm of the next cell. And so what we can see is that small molecules, primarily ions, are going to be capable of flowing through these gap junctions, and in doing so, they can spread a change in ion concentration within these cells kind of across a layer of cells or a group of these cells that are joined together by these gap junctions. A good example of gap junctions is going to be located within cardiac muscle cells. And so the cardiac muscle cells are individual muscle cells that are joined together. They have these gap junctions that's going to form a continuous path from the cytoplasm of one cell to the next cell, so that as a cardiac muscle cell is stimulated to contract, it's basically going to be triggered by an ion change in the ion concentration. This change in the ion concentration can spread through these gap junctions to neighboring cells and result in a coordinated and very forceful synchronous contraction of the cardiac muscle cell. And so the heart is pumping as a unit because of the presence of these gap junctions and the flow of ions through them from the cytoplasm of one muscle cell, cardiac muscle cell, to the next.